This is an introduction to Mendelian genetics. By the end of this presentation, you should be able to examine the lives and contributions of important scientists who effected major breakthroughs in our understanding of the natural and design world. In this specific PowerPoint, you should understand how Mendel did his experiments and what his results were. You will also become familiar with common genetics vocabulary such as alleles, dominance, recessive, phenotype, genotype, homozygous, and heterozygous. Before we begin, please recall that genetics is the science of heredity and the mechanism by which traits are passed from parents to offspring. Who was Gregor Mendel? Gregor Mendel was the son of peasant farmers. At the age of 21, Mendel joined the priesthood. Later, he went to the University of Vienna, where he learned how to study science through experimentation and how to use mathematics to explain natural events. Mendel then lived the rest of his life at a monastery, where he taught high school and cared for a garden. It was in this garden that he completed his important experiments. Because of Mendel's great interest in science, he was reading information on Darwin, who was studying the mechanisms of evolution. Mendel wanted to understand heredity. He decided to observe the garden pea plant. Modern genetics is based on Mendel's explanations for the patterns of heredity in garden pea plants. Before we discuss Mendel's experiments, let's learn more about the pea plant. Why was the pea plant such an excellent plant to study? Well, the first reason is that it has contrasting traits. In the study of heredity, physical features that are inherited are called characters. Several characters of the garden pea plant exist in two clearly different forms. They are contrasting traits. It is always either or. It is never and. What I mean by this is the color of the flower is either purple or white. It is never purple and white together. Another excellent trait of the garden pea plant is that it can self-pollinate. In garden pea plants, each flower contains both male and female reproductive parts. This arrangement allows the plant to self-pollinate or fertilize itself. Pea plants can also reproduce through cross-pollination. This process occurs when pollen from the flower of one plant is carried by insects or by other means to the flower of another plant. This allowed Mendel to control self-pollination and cross-pollination. The garden pea is a small plant that needs little care and matures quickly. And lastly, each plant produces many offspring, meaning many results can be compared for each type of cross. Recall that collecting repeated data is an important scientific method. If you look at this table, you will see the 7 contrasting traits of the garden pea plant. Flower color can be purple or white. Seed color can be yellow or green. Seed shape can be round or wrinkled. Pod color can be green or yellow. Pod shape can be smooth or bumpy. Flower position can be terminal or axial. Plant height can be tall or short. In sexual reproduction, gametes are produced. The male gamete is the sperm, and the female gamete is the egg. When the egg and sperm fuse or join together, this is called fertilization. In a flowering plant, sperm is located in the pollen, and the eggs are located in the ovums. After fertilization, seeds form, which in this case would be the peas of the plant. Ovules are located in the carpel. Peas can be self-fertilizing or cross-fertilizing. During self-fertilization, the pollen-carrying sperm lands on the egg-carrying carpel of the same plant. In order to allow for self-fertilization, Mendel would cover his plants. This prevented the sperm of another plant from getting onto the carpel of the plant he wanted to self-fertilize. Please take a moment to notice that the pollen is located on the anther, which is held up by the filament. Also notice that the carpel has three parts: the stigma, the style, and the ovary. The egg is located within the ovule, which is inside the ovary. The stigma has a sticky end, so when pollen touches it, it stays put. The pollen then travels down the style, into the ovary, to the egg, and fertilizes the egg. Flowers have adjusted nicely through evolution so that when pollen from another plant is unavailable, it can pollinate itself, therefore self-fertilizing. In order for Mendel to control his experiments, he needed to control which plants bred with which other plants. In order to do this, he first removed the pollen-bearing structures, anthers, of some plants to prevent self-fertilization. He then took the anthers from another flower and brushed the sperm, or the pollen, onto the original carpel to fertilize it. Using sperm from one plant to fertilize another plant is called cross-fertilization. Cross-fertilization is the process of breeding two different varieties of pea plant. The whole point of this is to create hybrid plants. Hybrid plants are the offspring of individuals with different traits. For example, if a pea plant with a purple flower were bred with a pea plant with a white flower, the offspring would be hybrid. Here you can see a visualization of what Mendel did to control which plants bred with which other plants. Observe that when the stamen is cut, it removes the pollen-bearing structures of the plant. After Mendel removed the pollen-bearing structures from the plant, he then used a paintbrush to transfer pollen from another plant onto the carpel of the original plant. When Mendel did his experiments, he completed monohybrid crosses. This is a cross that is done to study one pair of contrasting traits. In the image, you can see that the contrasting trait is the color of the flower. Mendel's first experiments used monohybrid crosses only. So first, Mendel allowed plants that had each type of trait to self-pollinate for several generations. This process ensured that each plant always produced offspring of the same type. Such a plant is said to be true breeding for a given trait. For example, every time a true breeding plant that has purple flowers self-pollinates, its offspring will all have purple flowers. Mendel used true breeding plants as the first generation in his experiments. The first group of parents that are crossed in a breeding experiment are called the parental generation, or P generation. Mendel then crossed two P generation plants that had contrasting traits, such as purple flowers and white flowers. He called the offspring of the P generation the first filial generation, or the F1 generation. He recorded the number of F1 plants that had each trait. Mendel then allowed the F1 generation to self-pollinate and produce new offspring. He called this new generation of offspring the second filial generation, or the F2 generation. He recorded the number of F2 plants that had each trait. So to reiterate, the P1 generation is the pure breeding plants with offspring that look like itself due to generations of self-fertilization. The F1 generation is the offspring of the cross-fertilization between two true breeding pea plants. And the F2 generation is the offspring of the self-fertilization of two F1 generation plants. Something important to note is that cross-fertilization between two plants did not produce offspring with blending characteristics. For example, when he cross-fertilized a tall plant and a short plant, it did not produce a medium plant. And when he cross-fertilized a round pea plant with a wrinkled pea plant, it did not produce a semi-wrinkled pea. To Mendel's surprise, when he cross-fertilized two pea generation plants with different traits, he got 100% of the same kind of organism. For example, when he crossed a tall plant and a short plant, all of the offspring produced were tall. Another example, when he produced a round pea plant and a wrinkled pea plant, all of the resulting pea plants produced all round peas. Mendel wanted to know where the other trait went. Why were there no short plants? And why was there no wrinkled peas? This spurred Mendel to continue experimenting. After the results of his first experiment, he allowed the F1 generation to self-fertilize to produce the F2 generation to see what would happen. Remember that after Mendel cross-fertilized one trait with another trait, all of the resulting offspring had the same trait. So after he allowed the F1 generation to self-fertilize, he found that 75%, or 3/4, had one trait, and 25%, or 1/4, were the other trait. For example, when he crossed an F1 generation tall plant with another F1 generation tall plant, the results were that 3/4 were tall and 1/4 were short. Or if he crossed a round pea plant with another round pea plant from the F1 generation, 3/4 were round and 1/4 were wrinkled. He wondered how this was possible. At the very least, he knew that the original trait from the parent generation did not disappear. It was simply masked in the F1 generation. For example, the short trait did not disappear. It was simply masked by the tall trait. And the wrinkled trait also did not disappear. It again was masked by the round trait in the F1 generation. Finally, Mendel allowed the F2 generation to self-fertilize. He found that 1/4, or 25%, that produced the wrinkled or short plant still produced the same trait, so he concluded that they were true breeding. The 3/4, or 75%, that had produced the round or tall trait in the plant was a little more complicated. He found that 1/3 were true breeding for the same characteristic, tall or round. He then found that 2/3 were not true breeding and produced the 3/4 and 1/4 ratio. Mendel interpreted his results in the following ways. He decided that traits were controlled by hereditary factors. In other words, each generation got their traits from the generation before. He also concluded that those factors occurred in pairs. He called those factors alleles. During reproduction, each parent contributes one of those alleles. For example, when he cross-fertilized a tall pea plant with a short pea plant, the offspring inherited an allele from each parent, a tall allele and a short allele. When an individual has two of the same factors for a trait, they are said to be homozygous, homo meaning the same. When an individual has two different factors for a trait, they are said to be heterozygous, hetero being different. For example, when a pea plant has two alleles that both code for being tall, it is homozygous because both alleles represent the same trait. If a pea plant inherits two alleles for being short, it is still considered to be homozygous because both of the alleles code for the same trait. On the other hand, if the plant inherits one allele for being tall and one allele for being short, it is said to be heterozygous because the two alleles are different. Mendel also deemed that one trait was dominant and one trait was recessive. Recall in the F1 generation that one of the traits was completely masked. This is because when the dominant trait is present, it is always shown. For example, plants are always tall when they possess the tall allele. On the other hand, recessive traits are only shown when both alleles present are for the recessive trait. For example, the plants are only short when they possess two short alleles. And finally, Mendel called the way a plant looks its phenotype. He also said that the actual alleles the plant will have are its genotype.